biotic, something that is external from the body, right? So the pharmacists among you would be able to be the matatu drivers, because pharmacy is just about delivery. And the pharmacologists around in this class would then look at the pharmacodynamics, mode of action, how this xenobiotic is interacting with the body and then eliciting responses. And these responses would be therapeutic responses. Sometimes they would also be toxicological responses. Yeah, and then of course you get the diagnostician, who is your medical doctor, who diagnoses the phenotype, if you like, and then you have these tools with you. And so part of this means that we need to know the target, right? So where are we sending this missile to? Right? Is this a place where Osama bin Laden is, or is it somebody else? And then we need to validate whether actually this person is there or not. Is this disease there or not? Does the target actually respond? Right. So let's go on. So this we can do through use of very complex, very modern tools. And actually now increasingly, we are using artificial intelligence, isn't it? Because it stores a lot of data. It's able to have algorithms which can screen a lot of these targets and actually be able to tell us that, well, if you design a drug in this kind of way, you're going to get better interactions or not. Yeah, so that's why people now talk about in silico drug discovery. So a lot of that is just people sitting behind a computer and doing that. But you can't substitute the experimental side. So once you've done all this computational modeling and bioinformatics, you will still need to do the work, the benchtop work, as it were. And somebody said that the only means of validating a target is when the drug works in the species of choice. So if you are wanting to treat something in a dog, it is only when that drug works in the dog that you can say, hey, we actually have validated the target. If you want to treat something in a human being, then it's only at that point that you would be able to say, okay, we have completely validated this system. Okay, so let's maybe think about drug discovery. And I see a lot of people here. I just want to ask Judith. Judith, you can just unmute, and Gilbert. So I'm not a lecturer, I like people to be, yeah, and yeah, Judith and maybe Gilbert. Is Judith there? Maybe she can just unmute. Maybe Faith, I'm seeing Faith and Fiona, you can also unmute. And Moses, are they able to unmute? Okay, Fiona. Hello, how are you?
I'm fine.
Yeah, so so what are you studying? Are you studying or you working? Are you a professor?
Pardon?
Are you studying or you're working?
I'm studying
Okay, what are you studying?
veterinary medicine.
Oh, okay, that's great. So where do drugs come from in veterinary, from the ones that you use in veterinary medicine? Where do they come from?
Pardon?
Where do drugs come from?
Okay, they come from different companies.
Okay. Where do those companies get them?
Some of them manufacture, some of them import.
Okay. All right. Thank you very much, Fiona. Then I had Gilbert. Or was it Fiona? Thank you. Right. There was somebody else. Yes. No. Moses. I don't know. Okay.
Yes.
So
Yes, sir.
yeah. Yeah. What's your role? What are you doing?
Yeah, I'm studying and at the same time I'm working at Masai Mara University.
Oh, okay. What are you studying?
I'm studying Master of Science in Medicinal Plant Research and Drug Development.
Oh, wow. Okay. So where do drugs come from?
Yeah, drugs sometimes they can be maybe from plants, like maybe I'm talking about the herbal medicines. Maybe other from pharmaceuticals companies.
Okay, that's fine. Thank you very much. You can mute. So the issue is, it's like asking a young person who lives in Nairobi in a city, where does milk come from? And a lot of the time they'll tell you that it comes from a supermarket of some sort, right? But actually drugs come from mostly natural sources, believe it or not. So we have what are called botanically derived medicines. Some of the most important drugs, if you think about morphine, right? If you think about aspirin, right? Just if you think about a lot of anti-cancer drugs, those are coming from plants, from botanical sources. And then we get plants, drugs that come from microorganisms. So penicillin is a good example. Even the lipid-lowering drugs, atorvastatin, it actually tracks its history from fungi, right? And then we have marine-derived compounds. Some of these neurological drugs come from there. We even have human-derived compounds, by the way. A lot of the so-called new medicines, like the biological biosimilar medicines, hormones, growth hormones, erythropoietins, right? Those actually come from humans. We have food-derived compounds. So let's not forget that things like vitamins and minerals are actually drugs as well. Then we have the new ones, right? Recombinant proteins. But these are really just, we are now applying technology to a lot of these ones, the basic ones. So in nature gives us a lot of drugs, right? So this is something that I will not talk about this, but this is something always to keep in focus when you're thinking about drug development. Obviously, you have to select the condition you would like to treat. You need to understand that condition and identify the drug target. So if it's a typical one, for instance, if it's cancer, what in the cell cycle are you thinking of interfering with? Are you looking for an antimitotic agent, for instance? If it's diabetes, which part of metabolism are you interfering with? Because you can interfere with the way that sugars are absorbed. So you have a drug such as acarbose, or drugs that are based on soluble fibers, right? And then you could have a drug that interferes with insulin. In this case, it's insulinomimetic drug. So it's a drug that acts as if it's insulin. The body kind of recognizes it insulin, right? And then you can have drugs that reduce sometimes or increase the sensitivity of insulin. So you need to understand that. But of course, and this is what we'll talk briefly about. You know, there's a lot of chemistry that goes on. And that's why for a lot of you who are in natural products chemistry, when I went into natural products chemistry about 25 years ago, I was so excited. I thought I was going to discover a drug. But there's a difference between the chemical, the constituents in a plant and a drug. And there's a world of difference. There are a lot of things that you need to do in between, right? And we will have time, hopefully, to talk about that briefly, right? Part of that is looking at structure-activity relationships and then optimizing the lead, as we call it, using DMPK techniques. And then, of course, you will go through all of these things, which today I will not talk about. Right. So let's then give you some examples of plants and drugs. And maybe some of you don't know this, but quinine, for instance, chloroquine. So chloroquine actually comes from a plant, but it came from a plant through quinine. So it is an improved form of chloroquine, right? The same with artemisinin. Right now, we are talking, artemisinin is a big thing for malaria treatment, even to some extent for COVID. But again, artemether is an ether, right, of artemisinin, and it comes from a plant. But now when it's in that vial, you don't even think that it's a plant anymore, right? Then when we look at some of the most important painkillers, I mean, we still cannot find drugs that are better at killing pain than morphine and related compounds, right? The opioids. These are from medicinal. These are from plants, and which is why there was a war in Afghanistan, as you may know, because Afghanistan is one of the major sources of opium. Not just for the opium trade, even though people want you to believe that, but also for pharmaceuticals. And animals, I think we've talked a little bit about this. So frogs, this is a frog that gave us epipatidine. Unfortunately, this proved to be very toxic, but this is interesting, right? There is a venomous snake, a Brazilian pit viper, which has given us the eels of this world, ACE inhibitors. So when you look at captopril and all the other drugs that have come into that class of antihypertensives, those actually, interestingly, come from a snake, right? Of course, if we had just milked this snake of this teprotype, which is a very complex compound, you can go look it up, it would have been highly toxic. So what we then do is, using drug design science, we are then able to increase the drug's benefit, the drug's efficacy, and reduce the drug's toxicity. Right, from the microbes. So microbial sources are known for giving us antibiotics. So pretty much all the antibiotics that we have in our armamentarium are from fungus, actually. So Penicillium notatum gave us penicillins. It then gave us the cephalosporins that you guys use today, meropenem. Pretty much any of these drugs that form the core of antibiotics. And that's why you're interested in antibiotic resistance. But also, surprisingly, fungi have also given us this blockbuster of drugs called lipid lowering drugs, isn't it? Lovastatin, which lovastatin was then modified into atorvastatin and into all the statins that you can think of. And this is actually from the oyster mushroom. So that's why some people say if you include mushroom in your diet, then you will be able to manage cholesterol. Right? Not much from the computers, interestingly, but we still have an antiviral, oseltamivir and related. And you will see here we have an antihypertensive drug, aliskiren, which not a lot of us know. Yeah, a lot of our antihypertensives actually are also from plants, the ones that we use in Africa anyway, from Madagascar and periwinkle. We can have forgetting now the specific one. What's missing out of that list? The things we talked about, the things that actually come from human beings and primate animals. So if you think about enzymes, insulin originally came from dogs. They used to actually get it from dogs, and then eventually it's now from a technology, recombinant technologies. Then, as you know, we have things like enzymes that we use a lot of. We have antibodies, and increasingly very complex immunotherapies. Okay, so testing. So once we have a lead, or we think, okay, we have something, how do we test these things? Cecilia? I've got Molens and Olusola. What do you guys think? How can we test these things? Cecilia, you can unmute. Olusola? Steven, Sharon, anybody?
Hello.
Hello,
I'm
how are you, Steven?
I'm fine, sir. How are you?
I'm all right. So where are you based?
I am based in, I'm a Ugandan, but I'm now based in Nigeria.
Oh, wow. You are
At the
well
Pan-African
traveled.
University of Life and Earth Sciences.
Okay, that's fine. So let's say that you have something that you think works. What do you do in terms of testing?
So what you do to test either for the efficacy or the toxicity of these drugs? Is that
Yes.
what you're asking?
Yes, yeah, that's what I'm asking.
Okay, they are what we call in vitro studies.
Mm-hmm.
And these in vitro studies are designed basing on what the drug is designed to treat. For example, if the drug that you found out is to treat maybe a bacterial infection.
Mm-hmm.
So you test that compound or that drug against that those specific bacteria that you want, that cause the disease that you want to use that drug to treat.
Okay, that's good.
Yes.
So in other words, sorry, go on.
You can also go ahead and do in vivo studies to test for the toxicity, determining the LDC 50, the EC 50, the LD10, to know how toxic that drug might be.
Okay, that's good. So what you're saying is that your assay has to be appropriate, right?
Yes.
Okay, it's got to be appropriate for the disease. For the disease, okay? Right?
Yes.
Anybody else? So an assay also has to be cheap. Okay, let's call it affordable. Because drug discovery is very expensive. Right? It has to show, it has to be reliable. Right? To be able to show repeatability. Yeah, so this is where we start talking about validation of the assay. Okay, and it has to be also appropriate for the species. For the species. So there are a lot of things that an assay has to be. So if, for instance, you are obviously it's got to be ethical, right? Because the most appropriate assay would be to test in the human being, right? But that would be highly unethical because we do not know the toxicity of whatever it is that we might be testing. Okay, so these are things for you to think about. Sorry. Okay, so we are, here now. Okay, now one of the most important assays that has come up over the years is actually the Zebrafish model, right? Quite fairly expensive, not easy, but it's because it's a vertebrate, it actually mimics a lot of vertebrates as well, right? So, and because it's an in vivo cell model rather than just cells that are isolated and they are in a petri dish, as it were. Okay, so it's actually it's got genes that are very close to the human being in the sense that it's about 70% confluence there. It's tough, so it can regrow. So it's actually very good for wound healing because you can wound this little fish and treat it, and there's regeneration. We've actually got a paper that should be coming out soon where we were looking at validating traditional medicines that are used for wound healing. It can also do a lot of things, like through genetic modification, you can make zebra glow in the dark in different colors. So you could use it for epilepsy. People are using it even for neurodegenerative conditions, and of course for cancer. It's fast growing. You can do these assays within a week and you're done. And then it's a vertebrate. So this is actually very useful. All right, setting up is a bit of a problem, but it's actually a really, really good and robust assay for testing a whole array of things from diabetes to cancer, to wound healing, to even, as we said, epilepsy. Okay. Then, so once you have this thing and you have tested it and seen, okay, it actually works, what you then need to do is to get into drug design. Because as I said, the compound that is active is not necessarily the ideal compound. It can be toxic. So when we think about rational drug design, right, we focus on what is called the pharmacophile. So in other words, if it's a single compound, and in this case, I'm talking about single compounds, there's a part of that compound which is what is responsible for the pharmacological effects, right? And then we can modify it. So it's like, think about a car, right? what is responsible for the movement of a car, that's why we call it a motor car, is the engine, which has a motor on it. But we can change that car, we can change the shape of the body, right? We can make it into a bus, we can make it into a big matatu, we can make it into a small car. As long as we don't interfere with the motor, with the engine part of it, it's still going to get us from A to B. And this is kind of how to think about it. So then what we would do with this pharmacophore is we would modify it, dress it up into what we call analogues. And these analogues, we are making analogues because we are trying to influence the potency of the compound, the spectrum of the compound. So if it's an antibiotic, we want it to be broad or we want it to be narrow, We're trying to influence the pharmacokinetics of this. In other words, the movement of the drug. That we can do as well. And we want to influence the formulation because there's some compounds, like insulin is a very good example. You can't have insulin by swallowing it by mouth, because then it's an enzyme, so it just breaks down. It's a protein, it just breaks down, right? But increasingly, what they're doing is they're coming up with analogues of insulin, right? And those analogues, it is hoped that we now have analogues which can be inhaled, so they are absorbed directly through the nasal passage, right? Great innovations around that. The analogues can also have improved solubility, which then impacts on dosing regimen as well, right? And then, of course, bioavailability, which is what all of you as pharmacologists would want to have. Down here, we want our analogues to be less toxic, and we want our analogues not to be vulnerable to resistance, development of resistance. So, for instance, if you have the penicillins, right? Certain enzymes will break them down. Beta-lactamase enzymes will break them down. But if you modify that structure, you might then reduce this breakdown, which actually the phenotype of that is drug resistance. So what we really want to do as drug engineers, call it that, is what I call engineering the great escape. So we want you to take a dose, and that dose should be able to survive metabolism in the gut, should be able to survive metabolism in the liver, and be bioavailable, get to the site of action that we want it to get to, do what it's got to do, and get out of the body, right? So, I mean, it's a miracle that this is what happens with drugs. That's how wonderful the human body has been made. And there are different ways. So from a chemistry point of view, you can do lots of different things. Here's a little case study of penicillins, right? So this is a pharmacophore of penicillins, right? The lactam part. It also happens to be the part where the enzymes clips, these enzymes. But this is the part you should not negotiate. So the non-negotiables are around this ring. If you change this ring, you don't have penicillin anymore. What are the negotiables? You can change the sulfur here. You can change the side chain around this. And when you do that, you can influence different things to that, right? So you can influence how it's taken. The first penicillins could only be injected, as you know. Now we've moved from injectable penicillins and we moved to ampicillin, which was taken 4 times a day. Now we've moved to amoxicillin, which is taken 3 times a day. And we've moved to amoxicillin with clavulinic acid, which sometimes can be taken twice a day, but is also more robust, less resistant to breakdown by beta-lactams. Case study number 1, done. Now, let's talk about drug discovery from traditional medicine, and that's case study number 2. And this is really based on what we call reverse pharmacology. But maybe is there a question at this point? Because I'm going pretty fast. I should finish in the next 10 minutes if it's okay. I don't know. Yeah, if there's no question, I will move on. Right. What is reverse pharmacology? So the normal pharmacology that we know is there's target selection, and then we get a lead compound. After testing many compounds, we then get a lead compound, 1, 2, or 5. And then we do molecular target identification. So this is now at the molecular level. And then out of that, we then get this lead compound and we optimize it for, that's where we talk about drug metabolism, pharmacokinetics. So we are now optimizing it to be metabolized, within a certain period of time. We modify it to be transported in some way, to be long-acting, short-acting, all of those things. Once we do that, we get into preclinical trials where we will test this in animals, rodents being the animal of choice, and then increasingly we might get into non-human primates, right? And then after that, we get into first-in-man trials. And these are really first-in-man because we don't use women, right? Because of reproductive toxicity issues. So we actually put this into male participants. And then increasingly, we will get to a point where we go through phase 1, phase 2, phase 3, and then we get into phase 4. So the trial of drugs never ends. I hear people say they are not taking vaccines because they are in a clinical trial. Well, I've got bad news for you. When you take paracetamol, when you take an antibiotic, you're still in a clinical trial. It's just called a phase 4 clinical trial, because there are actually some drugs that have been used for years and years, and then they get pulled off the market because they find, well, maybe these drugs now have problems, right? And recently they've pulled some very popular drugs off the market. Ranitidine, for instance, is not on the market anymore because there were things happening within the manufacturing of ranitidine. Then there's losartan and related products. There have been issues around that, right? But that, be that as may, that is classical pharmacology or forward pharmacology. Now, what is reverse pharmacology? Well, reverse pharmacology is what's happening right now with traditional medicine, because people are already using the products. They've been using these products for many years, many generations, right? But now we want this to know whether these products are being rationally used, whether they are efficacious, whether they are safe. So what do? Well, we are already in a trial, if you like. So people are already in trials. And normally we talk about observational studies. So we will look at observational studies, and we might then people continue to use the products. We might then start to try and put them into boxes, right? And then we try to understand, why does the product work? People are using it, but why does it work? That's mechanistic preclinical trials. So in this case, we would take this product, put it into animal studies and try to see. Then we might look at safety studies. But the fact that people are using these products surely should tell us that these products are generally safe, right? Then we might do molecular pharmacological studies and larger clinical trials. So that is reverse pharmacology. And there's a little thing that I put in here. So if you look at a plant that's being used for traditional knowledge, we can look at experimental observations. We can also look at clinical experiences. These clinical experiences could be of traditional healers and must include traditional healers. So if I work a lot with traditional healers, and we always say you need to keep a record of the outcomes, because then it's going to help to get the drug to market, which now is, well, in a different way, in a pharmaceutical way. Okay, so quick one, case 2, this is Godway's Combretaceae. You will see it. You will see it anywhere in Africa. There are over 250 species of Combretum, 250 of Terminalia. So it's very typical like this. Fossa maras, as we call it. This is another picture of it. This is my grandmother. So this was a plant that I fell in love with for my PhD. What is it used for? Abdominal disorders, hematuria. It's used to fatten babies. Presumably means that it's got anti-helmintic properties. It's used for infertility. Presumably, it may be that it's got antibacterial activity against the causative agents of STIs, because some people become infertile as a result of this. Syphilis, for instance. Sore throat, presumably it has antibacterial activity against maybe streptococcus, right? Dysmenorrhea. In India, it's actually used for congestive cardiac failure. The nut is considered toxic, right? But we know that what is toxic is also medicinal. So this is what we did, extracted all these things, and then came up with the compounds. And some of you might know these are flavonoids. This is a unique compound called combretastatin. You find similar compounds actually in grapeseed. This is pentacyclic triterpene. Okay, so very interesting compounds. This is a test in an in vitro assay, and you can see there's activity here. So you'll be able to say, okay, this is active, this is active. Because you guys are mostly antimicrobial people, these then are the compounds that you get. So we've got activity against Williopsis, Meraki, Candida. We've got Candida albicans, staph, TB, Mycobacterium fortuita, which is the safe one that you can use, Listeria. So you have actually, in a sense, validated that this plant works in vitro. You can see the concentrations are nice concentrations. And you can see there's structure-activity relationship. So as you modify, and this, by the way, I didn't modify it chemically. These are compounds. So as you modify the structure, remove the sugars here, you're actually getting potency as you go down this way. So this is a fantastic case study of structure-activity relationships. And this is what it actually looks like, the 3D model. And from here, you might then be able to actually do some docking experiments. So this is your in silico part of the study. What's the next stage? It would be to re-isolate the compounds. You can't synthesize these compounds, Nature synthesizes the compounds better than you can. The only thing you can do is semi-synthesize them. Then you can, in this case, we wanted to look at TB. So you can then retest on fast-growing strains because you can't really work with TB on the bench top. And TB takes a long time to grow. So you work with fast growing strains and you see whether there's good activity or not. And then you can go into making analogues first. Then using those analogues, you then go into the MTB assay itself. Okay, this is just, the slides were mixed up a bit. So this is just how they went about with the penicillins. I apologize there. Okay. Right, so that's it. Thank you. We have some books. So we have Veterinarians. Here's a book that we put together about ethnobotanical medicine. Actually, there's a contribution from someone who was at ICP there, right? So in Africa, botanical medicines in veterinary practice are probably the most important interventions because veterinary pharmaceutical products are just very expensive. So here's a book. I think you can look this up online or go to your library. This is our most recent book. We continue to say that there is room for traditional and indigenous knowledge in the here and now. And COVID has shown that more than ever. Thank you very much for listening.
Thank you so much, Professor Katerere. It's a pleasure having you. Thank you so much for the wonderful and very, very insightful presentation. It's always a pleasure to have a senior fellow in the house. So I just remembered, Prof. Katerere, in the midst of looking for Jimmy, I forgot to share about you. You know, it's good that our speakers know that we have someone very senior in the house. So I'll just share out your profile, and also if you love to maybe reach out to Professor Katerere, you can liaise with Jimmy and then he'll connect you. And thank you so much. So Prof. Katerere is, he did BPharm Hons and also holds a PhD. He's the research platform chair of pharmaceutical and biotech advancement in Africa at Tshwane, I hope I've spelled it correctly, Tshwane University of Technology. And he's also the chair of PharmaConnect Africa, a non-profit which is a premier convener of pharmaceutical industry in Africa. He also serves as a board member at the UI Global and the Association of African Medicinal Plant Studies. Clyde, Scotland, and he has worked in pharmaceutical and biotech practice and also research for the past 25 years in 4 countries and in 3 continents. And he has several inventions to his name. This is also an encourager to some of us in the session. You know, you can also do it, including Nutraceutical, Nicello, and COVID Connect app for using clinical trials and post-recovery. He has published over 50 journal articles and is the co-editor of 3 books, some of which he has just shared: Veterinary Botanical Medicines, published in 2010, Systems Analysis Approach for Complex Global Challenges, published in 2018, and Traditional and Indigenous Knowledge for the Modern Era, published in 2019. Prof. Katerere is also a member of committees such as the Global Health Supply Chain Consortium, which he co-founded by faculty at the University of Michigan and University of Southern California. He teaches and researches across pharmaceutical and biotech value chain, including product development for medicinal plants and clinical testing, substandard and falsified medicines and vaccines, and medicine governance. And he's a competitive Toastmaster, and he's a keen Rotarian and member of Pretoria Capital Club. Thank you. I think we share something on the Rotary Club. Prof. Katerere. It has been an honor having you for the session. We are really privileged and really honored to have you in this session, share some of the insights with us. And our hope is that we'll have more young people take up the good work that you have done and follow the same trajectory, so that at least we can also see whether we can also have Africa in the map on drug research and discovery. So I'll just read a question that was asked here by Stephen Kigozi. I think it was more of a request. Thank you so much for doing the good work you're doing. I have a special interest in microbiology and immunology. I'm interested in researching about how we can use medicinal plants to overcome antimicrobial resistance, which is currently a serious global public health challenge. Do you have any internship or mentorship programs where I can get on hands training in that regard? Maybe over to you, Prof.
Yeah, so no, not quite. I've moved out of this area of antimicrobial, but I mean, you can maybe look up University of Pretoria, they might have. It's just that you see that a lot of companies don't invest in antibiotics, because remember, with antibiotics, you only take a 5-day course, right? So from a business perspective, if you have to develop a drug for somebody with diabetes versus a drug for somebody with an infection, you go for the diabetes one because you're a lifelong patient. So those are the realities of it, right? But certainly, I think you can look at, I think there was a program in not King's College, but School of Pharmacy, the UK School of Pharmacy. Yeah, so if you look at that program, they might have fellowships where they were looking at combining plants with antibiotics. The rule of thumb, though, is that remember that plants are not attacked by bacteria. Plants are attacked by fungi, right? So if you're working with plants, you should be looking for antifungal compounds, right? If you're going to look for antibacterial compounds, you need to go and look for fungus as your source, or you can look at some of these aquatic kind of animal forms, because they are more likely to be attacked by bacteria. But that's actually a very, very important thing to look at. Yeah. Do not look for antibiotic compounds from plants. Look for antifungal compounds. Thank you.
Thank you so much, Prof. Maybe to complement. So as you can see, at the moment, this webinar and the ones that are coming after this, they are more on, it's a webinar series, and the aim is, it's just stage 1 of the program, the phase 1 of the program. So after this phase 1 of the program, we are working with various experts to see whether we can set up a FENT program for those students who will have shown much interest and kind of passion by attending this webinar series. So we'll create a FENT program for them. It's still in development, and we hope that we'll be able to link up with other organizations to kind of also try to have several placements and, you know, some attachment or internships for the people who will be engaged in the FENT program. So Stephen, you can just stay on hold. Maybe we have something good for something good that cooking, and yeah, we hope that at the end of the FENT program, at least we'll have some of the students who will be placed in these other institutions. Yeah, and so thank you so much. Unfortunately, our other speaker was out of power at his place. So maybe as we give him some time, we can just maybe share some questions. If you have some questions to Prof, you feel free to unmute, and yeah, or any other guidance that you require. You see, he has a wonderful experience, very deep expertise. So yeah, you can feel free to unmute and ask. Thank you.
Okay, I have a question. Hello.
Laban, yes, you can go ahead.
Okay, I have a question concerning this series of meetings. Okay, about the links. Will we be receiving the links like before the meeting day about this?
Okay, or about the program? Yeah, about the series.
Yeah.
Yeah. We'll be sharing the the links to the meetings. We have a group as you were sharing. I don't know whether you're in the group as you were sharing we also shared the link to join the group. So we'll just even the recording will share in that group, and it's from that group that we'll kind of select some of the students whom will engage in the defense program. Yeah. So at this juncture, I would love to hand over to Jimmy. Jimmy is the head of the program, maybe for something. So Jimmy, you can comment on something as I wait for speaker.
Okay. My name is Jimmy. I'm a student at Jomo Kenyatta University of Agriculture and Technology, Kenya. Actually, I'm so happy. Thank you so much, Professor. Actually, today we have learned a lot actually concerning the plants and the development of drugs from the plants. Actually, we have learned a lot. I think after this, the knowledge that actually you have acquired in your session, actually, we are going to utilize maybe to develop some of these antibiotics, because now if you can see that our main target, our main core is against this occurrence of antimicrobial resistance. Actually, we are so much grateful for this chance. Maybe we look up, maybe to have you maybe in more webinars as we come. Actually, we are so happy to interact with great intellects like you. We are so happy today. Maybe, for example, as we prepare for the further webinars, maybe up to on December, we'll keep on in contact with you. Actually, we would like to interact more on this. Secondly, I'd like to urge maybe my fellow students, actually, it's so important we participate in this action, in the activities against antimicrobial resistance. We should be researchers. Actually, as you can see, the rate of antimicrobial resistance, it's highly a very great challenge that is disturbing the health sector, global public health. At the same time, we should not be tired of getting, maybe, for example, getting into research to mitigate this health threat. Maybe before I go on, I would like to give a chance to those who may have some question to ask before we let in our second speaker. She has been facing some challenges in power connectivity, but hopefully will be in the next few minutes. I can just allow some of maybe a chance for some questions. Maybe to start with, I would like to ask a question, maybe for example to Professor, maybe. Actually, in Africa, you can see that research is actually the rate of research and discovery is very low as far as drug research and discovery is concerned. Actually, what might be contributing to this? Maybe, for example, to the low discovery. As you can see, we are highly over dependent over the developed countries to get into the, maybe, for example, for the drugs, antibiotics, and the vaccines. Actually, what are the challenges facing Africa as far as drug research, discovery, and innovation is concerned? Thank you.
Okay, yeah, and thanks, Jimmy. You were very persistent in trying to get me to your platform, so so I couldn't really disappoint you. So at the end of the day, it really boils down to resources, so research, as you know, costs money. Already people are asking around, scholarships. People, once you have a scholarship, you will need to think about purchasing of, because whoever is going to be hosting you, right? They are actually then purchasing reagents, for instance, which can be problematic. And it's easy enough to do these in vitro assays. Sometimes, they also cost money, but when you start getting into animal studies, that's extremely, extremely expensive. And then you get into human beings, if you ever do. So it boils down to the fact that we really don't have an ecosystem which is supportive of research. Maybe we have just too many priorities, which might sometimes be valid priorities, but sometimes not very good priorities. So we do need to create ecosystems of research, even within the university system itself, Because sometimes you find in the university, you're not promoted because you're publishing papers. Or if you're promoted because you're publishing papers, you don't get the time to actually do the research. You know, your boss is going to say, have you taken your classes? Have you marked? You know, where are the marks? They don't give you time to actually conduct research. So those are structural issues around why we are not moving. And the other thing is maybe collaboration. We are not collaborating enough. What I tend to find is everybody is trying to do the same thing. So we are sometimes competing against each other rather than actually trying to build areas of specialization. Which is why, for instance, I don't really do this kind of work anymore. I did this when I was still associated with the University of Pretoria, and I've shared on the group with my colleague at the University of Pretoria. So you'll see her on LinkedIn, and you can reach out to her. I've also shared on the group Professor Michael Henrik, who is at School of Pharmacy, University College London. He is an ethnobotanist. I don't know what sort of opportunities they would have for funding, but certainly worth looking at that. Thank you.
Thanks so much, Prof, for answering that question. Actually, as you can see, as just of answered, maybe for example, as you can see in Africa, actually, in terms of collaboration, actually, we are so poor in Africa. And I think maybe, as far as capital investment is concerned, to do research on antibiotics, research, drug development, and all those, actually, it is much expensive. So I think there's a lot that is needed, maybe, to bring in efforts to brainstorm. Actually, we should facilitate this research in terms of Africa. Maybe, for example, as time goes by, there will come a time where, maybe, for example, we will not be able to get enough that we can get, that we need. So actually, it is the high time that we challenge ourselves. What are the right steps that we should take, maybe to mitigate this problem of antimicrobial resistance? Because now, as you can see, through collaboration, we could be able to solve these challenges. We could be able to channel the funds together. We could be able to brainstorm, provide the necessary skills and knowledge. Actually, it is very important. Africa, it is so big, it is so large. It's so large. Actually, we are more than the problem that is facing us. So it is high time, actually, we should turn up and should activate our efforts towards these challenges that are facing generally Africa and also the globe at large. Maybe to give background information about this our program, the CS Africa. Actually, it's just a students registered organization that we do a lot of stewardship activities, research activities about antimicrobial resistance. But now, if you see, we are facing a lot of challenges when it comes to research. Maybe, for example, we might be able to have some ideas, some projects underway, but to facilitate them become a very big challenge. And that's where the students are facing the challenges. And also, it is high time we call upon any assistance from any willingness support, actually, to provide this support, actually, so that we can be able to facilitate our efforts as students. Maybe, for example, the universities that we're learning in, they're also not giving a lot of attention to us, the activities that we are doing. That become a very big challenge. The students become discouraged about what they are doing, but actually it's a very big challenge that is facing us. Actually, we should, through Professor Katerere, actually, I know you have interacted with a lot of these people, these researchers. You have traveled a long way in Africa. Actually, you could also assist us. You could be part of the team also, because antimicrobial resistance, it's a very big challenge, a very big problem that needs a lot of attention. Thank you so much. Maybe I can give out a chance to any other questions as we wait our next speaker to get in. Thank you.
All right. I will be exiting now, but it's good to meet with your group. Let's keep talking. And yeah, maybe we'll be able to work together in some form or shape. There are some antimicrobial stewardship groups that I know about in South Africa. And before COVID, I think there was a huge focus on this, but COVID just threw everything out. I suspect we're going to have even worse issues in this area because antibiotics are just being dished left, right, and center to manage COVID, unfortunately. But yeah, so we'll keep in touch, Jimmy, and then see. All right. So I wish you the best. I just have to prepare